Okay, so here we are in the herbal database, and we're going to dig into the intestinal mucosal barrier. If you're new to the herbal database, one way to find it is to use the search box, type in intestinal mucosal barrier, or you could use one of the menus. It's in the digestive system menu and also the immune system menu. All right, so we see it's a very complex, multi-layered system, and it has several components. There's the physical barrier. That's the mucus layer secreted by goblet cells, the intestinal epithelial cells, including enterocytes, goblet cells, Paneth cells, and M cells. Also, tight junctions between the epithelial cells. Now, we haven't gotten into this next part very much yet, but we will. There's a chemical barrier, which includes antimicrobial peptides secreted by those Paneth cells. Remember, those were down in the crypt. Bile and gastric acid, lysosome and other enzymes. There's an immune barrier, and that consists of the secreted immunoglobulin A, and we'll get into that more specifically in our next class. There are both the innate and adaptive immune cells in the lamina propria. Here's a diagram showing the lamina propria. If you'll recall, these are the villi in the small intestine. There you see the epithelial cells. The enterocytes is what they're called in the small intestine. This part is the crypt. The Paneth cells will be down here. And here is the circulatory system for the blood and lymph. And this orangish part is the lamina propria. Underneath that is a layer of muscle, and then submucosa down here in the blue. And here you see blood vessels, and I think that is to indicate lymph, but I'm not sure. Probably lymph. Then we also have a microbial barrier. That consists of our commensal bacteria that compete with pathogens. So while enterocytes are a crucial component of the intestinal mucosal barrier, they're just one part of a complex system that protects the body from harmful substances and regulates nutrient absorption. We spent some time looking at a diagram of epithelial cells, specifically the enterocytes. Those epithelial cells establish and maintain the barrier. In the skin, it's a tight, although not impermanent seal, but most mucosal epithelial cells form leaky barriers, and that's necessary to support universal functions that include fluid exchange, which occurs at nearly all mucosal surfaces, as well as essential tissue-specific functions. So the precise permeability characteristics of each barrier type differs based on the functions it supports. The ability of tight junctions to discriminate between and restrict passage of solutes based on size or size selectivity varies with the location in the intestine. Permeability to larger solutes decreases from the crypt up to the villus. In addition to these fixed differences, mucosal permeability of many tissues is adaptable and can be regulated in response to extracellular stimuli like nutrients, cytokines, and bacteria. To protect itself from uncontrolled inflammatory responses, the intestinal epithelium has developed mechanisms to restrain bacterial growth, limit direct contact with the bacteria, and prevent bacterial dissemination into underlying tissues. The mucosal barrier, which consists of only A single layer of epithelial cells is one of the most important components of the innate immune system, and all that separates the inside of the body from a very dirty outside environment. Thus, mucosal barrier functions a key component in the arsenal of defense mechanisms required to prevent infection and inflammation. The mucosal barrier functions maintained by several interrelated systems, including mucus secretion, chloride and water secretion, and that's going to be more down in the colon, and binding together of epithelial cells at their apical junctions. Remember, the epithelial cells are polar, meaning there's a top and a bottom. Apical means the top. That's the part that's sticking into the lumen. So those are bound up there at the top by tight junction proteins. And together they act as a gatekeeper of the mucosal barrier. Now, we talked a bit about what psychological stress does. So I'm going to pull this down. You'll find this part under intestinal mucosal barrier if you want to review it. So what does psychological stress do? It does disrupt the intestinal mucosal barrier. It alters tight junction protein expression. So there's a number of proteins, we won't get into that form the tight junction, and it can cause them to become more permeable. It impairs interleukin 22 signaling. We haven't gotten into IL-22 yet. It's a cytokine that maintains the mucosal barrier integrity. It increases intestinal permeability. Stress leads to intestinal hyperpermeability. We call it leaky gut, resulting in decreased transepithelial resistance and elevated paracellular and transcellular macromolecule permeability. So what does all that mean? Here's a diagram. These are just general epithelial cells, but it shows the different pathways. So the paracellular permeability pathway is when it goes in between the cells. Now, as you know, we've got the tight junctions and the adhesion junction and the desmosomes down here. So that's the paracellular pathway. And then transcellular permeability pathway. So there is a channel up here that glucose, for example, that's the only way glucose can get in, and it just goes down and comes out the other side. And if you recall from class, one of the problems is if there aren't tight junctions here, these channels can float around the cell and wind up in a weird place so that nothing can get in from the top, and everything's blocked. And then we have just this diffusion, I guess you would call it, where very small molecules can just go straight through the cell. And then, of course, psychological stress elevates stress hormones. Stress increases levels of corticotropin releasing factor in serum and intestinal tissues, which can mimic stress-induced intestinal dysfunctions. It also causes microbiota dysbiosis. Stress alters the gut microbiota composition, often leading to an expansion of potentially harmful bacteria, such as Enterobacteriaceae. It enhances pro-inflammatory signaling. It causes a shift in intestinal epithelial cell gene transcription, promoting pro-inflammatory, antimicrobial, and reactive oxygen species signaling pathways. Psychological stress impairs mucosal integrity. It disrupts the structural integrity of the mucosal layer, compromising its protective function. And there's increased bacterial translocation. It leads to elevated levels of lipopolysaccharide in the bloodstream. Lipopolysaccharides are an endotoxin that are released by certain pathogenic bacteria, and it causes a huge immune reaction, and also increased bacterial dissemination to other organs, such as the liver. Then we briefly touched on how mucosal barrier dysfunction can cause alterations that lead to chronic fatigue syndrome. And these two paragraphs in here will talk about that some. Now let's zip over to tight junctions. And here is a better diagram of what they look like. This is the tight junction right here. These are adhesion junctions, and these are desmosomes. We hear sometimes in leaky gut that the tight junction has been opened by zonulin, for example, but in truth, this never opens. What happens is I describe them as pores. There are areas where small molecules can pass through, and something causes those pores to expand, so larger molecules and even whole bacteria could potentially move through this. Then down here, these adhesion junctions, they exist to keep those cells linked up together so that they don't drift apart. That's why they're called adhesion junctions. And the desmosomes down here, that's a communication device, so the cells can talk to each other. These are the microvilli up here, and there's the nucleus. So if those tight junctions act as doorways that allow things in, what's the key that opens them? It turns out that one of them is called zonulin, and zonulin is one of our tiles. That's a protein that specifically targets and disrupts the intercellular adhesion molecules that are responsible for holding tight junctions together. Zonulin is released from a little, I guess you could call it a little tiny portal on the endothelial cell. Something has to trigger it to be released, and we'll get into that later. But once it's released, then it can open up. And again, remember, those tight junctions don't really open. It's just that there are pathways, I guess you could call them, or pores that expand. And that allows for some things like proteins to get into our body that should not. So there are some other things that can influence our tight junctions besides zonulin. For example, cytokines. If you recall, cytokines are chemical messengers of the immune system. There are pro-inflammatory cytokines and anti-inflammatory cytokines. Basically, they're telling the immune system to produce inflammation or to roll that inflammation back. There are bacterial toxins and metabolites. For example, I already mentioned lipopolysaccharides can get through. They can open it up. Short-chain fatty acids, however, are great for tightening those junctions. That's a tile that we have, and we'll be really focusing a lot on the short-chain fatty acids and how to produce those to have a healthy gut barrier. There are some dietary components that have an influence on those tight junctions, like prebiotics and certain omega-3 fatty acids. These are going to influence tight junction function. And then, of course, physical and psychological stress. We already talked about psychological stress. The physical actually can come from people who over-exercise, athletes, anyone who's doing heavy training. That can open them up. And there are certain medications and drugs, for example, laxatives and some antibiotics. They can have a temporary effect. Now, remember, when the tight junctions are opened via zonulin, they tend to stay open for about 48 hours, and they will automatically close back up. Zonulin, let's go over to zonulin here. Take a look at it, because it's not a bad guy. We need it, and let's see what it does for us. There are a lot of videos here that you can watch. Gliadin and bacteria are the two strongest stimuli to release zonulin. And then once zonulin is released, it communicates with our enterocytes to open the gate of the tight junctions. This all happens within 20 minutes or so. When the stimulus is removed, everything goes back to baseline in a very short amount of time. Okay, some questions here. Let's see. A lot of this, I'm going to leave it to you guys to come in here and read this yourself. Okay, this has to do with actually opening tight junctions. It explains how zonulin doesn't literally open tight junctions, but modulates their permeability through all of these different signaling mechanisms. How do bacteria pass through tight junctions? And I'll leave that to you to read, but as you see, bacteria can get through, and that's one of the problems with intestinal hyperpermeability. Bacteria is never supposed to get into the bloodstream. And here's about zonulin genetics. We'll skip that. All right, what's the main function? It regulates intestinal permeability. It acts as a molecular gatekeeper, modulating tight junctions between epithelial cells lining the intestinal wall, and it controls the passage of substances from the intestines into the bloodstream. So there's some stuff we do want to get into the bloodstream. It maintains intestinal barrier function when properly regulated. It ensures proper control of molecular and cellular traffic across the intestinal epithelium. It's involved in our innate immunity. It does play a role in protecting against microorganism colonization of the proximal intestine, contributing to innate immunity. It's involved in cell repair. It'll help to repair damaged cells in the intestinal lining. It regulates tolerance and immunity, together with GALT, the gut-associated lymphoid tissue. That's going to be a future class. And the neuroendocrine network. It helps control the equilibrium between tolerance and immunity to non-self antigens. It modulates epithelial and endothelial barriers. As far as regulating those zonulin levels, that's really important. If you've got chronically high zonulin, those gateways are going to be open longer than they should be. So it's regulated by bacterial exposure. Both pathogenic and commensal bacteria in the small intestine can trigger its release. And then we already talked about gluten exposure. That interacts with the intestinal epithelium, leading to zonulin release. Again, that's really a problem for people who are gluten sensitive and those with celiac disease. Those are two different things. Not so much with healthy people. And then its secretion depends on this adapter protein. That's hyperlinked, so when you come into the database, you can click on that and find out what that is. Also, environmental factors, stress, nutritional habit, other environmental impact can affect its levels by influencing the gut microbiome composition. Everything's linked together. And then in certain autoimmune conditions, for example, celiac and type 1 diabetes, zonulin can be upregulated. I'm not going to go through this because you guys are going to need to come in the database and read about these to find out what these are. Protease activated receptor 2, epidermal growth factor, those are both involved. So there are diseases that are associated with excess zonulin. Here's a list of them, and there's a lot. Autoimmune diseases like rheumatoid arthritis, type 1 diabetes, various cancers, various diseases of the nervous system, even schizophrenia. It can get into the serum. This part explains how it gets into our serum, and so there are tests. If you look over on the left, you'll see zonulin testing, and you can click on that, and that'll give you an idea of how much zonulin you're producing. There's some controversy, of course. It plays a role in colon cancer, and there are some inhibitors of zonulin. So if you take a zonulin test and find out that it's too high, there are some things that can inhibit it. For example, now that's a drug, but polyphenols. Certain plant-derived components like resveratrol, curcumin, and quercetin can inhibit it. Probiotics. They're not direct inhibitors, but they can positively impact intestinal barrier function and thereby indirectly contribute to zonulin regulation. And then, of course, the gluten-free diet for people who are sensitive. And then there's this AT1002. That's a zonulin-derived peptide. I don't know how you get a hold of that. But probiotics appear to be more effective at inhibiting zonulin in certain health conditions. For example, people that have metabolic disorders, people with cirrhosis, folks with inflammatory conditions, if they have colorectal cancer, or exercise-induced stress. That's where probiotics are going to be a real good go-to. Let's see if there's anything else we need to get into now. You can find out how zonulin interacts with other immune molecules in the gut. And then we've got a lot of research articles here that describe zonulin's role in arthritis, asthma, cardiovascular disease, COVID, the gut-brain axis, so on and so forth. So that's, I think, enough of an introduction to zonulin, so let's move on. The last thing we talked about in class was yogurt. One of the questions that I had was, assuming that we have sufficient stomach acid, it's killing off the bacteria before it ever gets into the gut. And if that's happening, how in the world are the beneficial bacteria in yogurt surviving? So I did some research on that, and we're going to dig into it now. So again, some videos here, and here's my question. How does yogurt benefit the intestinal mucosal barrier, since stomach acid kills bacteria? And this is what I found. While it does kill many bacteria, yogurt can still benefit the intestinal mucosal barrier through several mechanisms. First of all, acid resistance. Many probiotic strains in yogurt, particularly lactobacillus species, are naturally acid resistant and can survive the harsh stomach environment. Then there's also a protective food matrix. It provides a protective environment for bacteria, helping shield them from stomach acid during transit. Also, quantity matters. Yogurt contains high concentrations of beneficial bacteria, increasing the likelihood that some will survive and reach the intestines. Next, transient colonization. Even though probiotic bacteria don't permanently colonize the gut, they can transiently populate the intestine and confer benefits. Improved barrier function. That's what we're looking for. Regular yogurt consumption, at least 2 cups a week, has been associated with lower plasma-soluble CD14 concentrations, suggesting improved gut barrier function. Now I'll point out, you see the numbers here, those are links to studies and research articles that you can read that back all this stuff up. Tight junction regulation. Certain yogurt starter strains, such as Lactobacillus bulgaricus 2038 and S. thermophilus 1131, can ameliorate physical barrier dysfunction. So when you look at your yogurts, look and see if they're showing those. And this is interesting, honey enhancement. Adding honey to yogurt can help protect the beneficial bacteria during digestion, particularly in the intestinal phase, allowing more live bacteria to reach the gut. Indirect effects. Even if some bacteria don't survive, their metabolites and cell components can still have beneficial effects on gut health. All right, so we've got quite a bit on yogurt here. How does yogurt improve gut barrier function? First of all, of course, with the live bacteria. Look for strains like Lactobacillus is almost always there, but see if, hopefully it has bifidobacterium in it. And bifidobacteria is one of our new tiles. Again, that's something we're going to dig into more later. Those can temporarily populate the gut and confer health benefits. It reduces plasma-soluble CD14. That's 2 cups of yogurt a week. Mucin production. Mucin is one of our tiles. Animal studies suggest that a peptide derived from the beta-casein in yogurt increases the production of mucin, and that's an essential component of the protective mucus layer lining in the intestine. Mucin we'll get into next week. Modulation of gut microbiota. It can beneficially alter the composition and function of the gut microbiota. It strengthens epithelial defense. Certain probiotics in yogurt fortify the intestinal barrier by strengthening epithelial defense functions. It has anti-inflammatory effects, and it regulates tight junctions. Some yogurt starter strains can ameliorate physical barrier dysfunction by activating AMPK and regulating tight junctions. Again, if you don't know what some of these things mean, you can look them up using the search function. You could look up, for example, AMPK in the search box, find out what that is. Okay, then of course we get into what are the main probiotic strains that benefit gut health. I'm not going to go through that. It impacts the diversity of gut microbiota, and here's more details on how beta-casein helps the barrier function. As usual, there are a lot of research articles, clinical studies, etc. in here under articles and studies. I'm just going to scroll through so you can kind of get an idea of what yogurt can do. Here's immune system effects. Plays a role in decreasing Th17, that's good. It reduces postprandial, that means after eating a meal. It reduces postprandial inflammation and markers of endotoxin exposure in healthy people. Let's see. Okay, that's enough there. And then, of course, we've got all kinds of yogurt recipes, so on and so forth. So that's where we ended, and I hope to see all of you guys in class next week.